Yes. Good evening to all the participants. Welcome to the 11th session of this certificate course. So the 11th session is on T cell epitope prediction. Before going to the T cell epitope prediction, can we recap the last session contents? So if you see the previous session, that is 10th session, we discussed something about basics of concepts of reverse vaccinology and various immunological databases. Okay, so we discussed about immunoinformatics and we discussed about reverse vaccinology. I think I didn't take you much about the immunological databases that we'll be discussing it on today, along with the T cell epitope vaccine design. So, can I ask you a few questions on today, like to recap the last session contents? The first question is like, what is the first vaccine uh, developed from reverse vaccinology? Vaccinology are of two types: clinical vaccinology or reverse vaccinology. Very good. Meningococcus B vaccine, isn't it? For treatment of what disease? Can I know the disease name? The vaccine name is correct. You're right. How about the disease name? Very good. It is bacterial meningitis. But, so for this, we have used a series of screening procedures. Okay, just like that, we can't able to even whatever we get from the competition, we can't take it as finalized one. One has to check whether it is feasible during the recombinant technology. Okay, and also one has to check whether how much they can able whether they can able to go for pilot scale production or not. So based on this only, we can screen the vaccine candidates. Okay. So I think we have discussed on yesterday related to this. And then we also discussed something about immunoinformatics. Immunoinformatics is nothing but computational immunology. Okay, so that's what we call it as an immunoinformatics. Here you have can find in various applications like producing, even designing the antibodies. Antibody design is also coming under immunoinformatics, and you can also design the mRNA. You can screen the mRNA. Okay, you can come across peptide-based vaccines. Okay, yeah. So all these things, okay, comes under the immunoinformatics. So now let me take you to the immunological databases and a T cell epitope based vaccine design. So before going to the T cell epitope, so I can show you some databases. The first database what I'm going to show is the IADB. This is the largest database. Okay, so where I can find tools for predicting B cell epitope and T cell epitope. Other than these epitopes, so we can also go for epitope analysis. Okay, after prediction of epitopes, if you want to find, okay, how about the epitopes are covered and the specific alleles, like covering the certain people? I have mentioned you in the previous session that even you can specifically design SARS-CoV-2 for a Tamil Nadu population, especially for the Nadar community. Okay, all these things you can able to screen. Okay, you can go for uh, the specific community, the specific epidemiology, etc., from this population coverage and conservation across antigens. And also, these are other kind of tools available in IEDB. But the main databases you can find, okay, we generally go for search of epitopes from here. And not only you can search the epitopes, the types of epitopes, but also you can search for various diseases. Epitopes can be a linear or discontinuous. We have seen on yesterday for B cell, but other than that, we can also able to predict non-peptidic epitopes. Okay, so there are a lot of types even in epitopes, and yeah, even the sources they are not. They didn't just limit up to the animals. They also provided sufficient data for the plants. Okay, so even if you are dealing with plant viruses, if you want to come up with a kind of remedy for the plant viruses, even you can use this database. Okay, so not only it is developed for human, it is developed for almost all the animals. So generally, those are doing the veterinary practice, they can also use this kind of a database to come up with a kind of a vaccine. Okay. So now let me go ahead on the T cell epitope prediction. Then I can take you to the how to use this tool. There are plenty of tools in immunoinformatics. Okay, so this is the major thing. At least first you should know how to use this tool. Then we can go with the other tools also. Okay. So now I take you to the presentation T cell epitope prediction. I told you if you see the major classes of epitope prediction, one major classification is T cell epitope, and the other classification is the B cell epitope. If you see the case study of Nipah virus, okay, so if you want to bring a vaccine for the Nipah virus, so one has to bring only for the efficient vaccine to treat for the Nipah virus is the B cell epitope because the antibodies play a major role in curbing that infection. However, okay, if you want to try with a few more viruses, of course, T cell epitope plays a major role. So in that case, it can also design accordingly. Now, if you see the T cell epitope, on yesterday we have seen two types of cells, cytotoxic and helper T cells. So for these cells, okay, you have a different kind of MHC molecules in APC is going to interact. Okay, for example, CD4 T cell interacts with the MHC2 and CD8 T cell interacts with the MHC1. Okay, so the length of the peptide varies. Okay, the length will be around 13 amino acid, which is greater than the CD8 T cell epitope, which is just uh, uh, 9 amino acid in length. Okay, actually it is in range, but if you see even in Coursera courses developed by the John Hopkins University, they prefer to use this kind of a value, so you can fix it up for your research. We already discussed something about how the B cell receptor looks like and T cell receptor. More about this, you can study from the immunology textbooks. Okay, we are just bothered about the T cell receptor recognition, this part alone. Okay, if you see the B cell receptor, you have two, just like an antibody, you have two different recognition receptors. Okay, here only one. Okay, you have a kind of a domain like alpha and beta. Okay, so above that you have this kind of a T cell receptor recognition. So if you see this T cell receptor recognition, so this is what? Okay, so this is going to the V alpha and V beta. Okay, which is try to interact. Okay, no. If you see the T cell receptor, uh, this is what actually V alpha and V beta is try to interact with the alpha 1 and alpha 2 of MHC1. Okay, in MHC1, if you see uh, the paratopic region of the MHC1, you can able to see, no, actually whatever is going to have, okay, this is an epitope and this is what the paratopic region. In T cell, you can call it as a paratope just because this antigen is carried forward by the MHC1 molecule. Now we call this region as the epitopic region. Okay, so in the paratopic region, V alpha, V beta. So you have just this alpha and beta, whatever I showed there. Okay, so one is constant domains, and if these are the constant domains at the bottom, and variable regions alpha and beta are at the top. Okay, so two different regions are there. Similarly, for the B cell receptor, more about this we'll see on the next session. So we just focus on the T cell receptor alone. So I think we have already seen this figure, and now yesterday also explained you in dendritic cells there are so plenty of surface receptor molecules around 160. I have identified 162 molecules. I used actually to come up with a best adjuvant for treatment of specific disease. So I tried docking of multiple plant-based adjuvants with these 162 receptors, and based on the scores, okay, so I can even able to predict exactly the like a plant-based adjuvant A can be used for what disease, plant-based adjuvant B can be used for what disease. Okay, all these things you can able to get from the binding affinity scores, and even DC T cell interaction. Okay, so not only we bother about to get T cell activated, even the dendritic cell has to be activated so that even the dendritic cell populations can also be increased. So you know the dendritic cells or not just the antigen presenting cells, these are professional antigen presenting cells. Okay, dendritic cells alone we call this as professional antigen presenting cells. Like dendritic cells, we also have macrophages, T cells. These are called as antigen presenting cells. Generally, why we call this as antigen presenting cells? So these cells, okay, has two different molecules on the surface. That is MHC1 or MHC2. This is the major receptor we concern on. Okay, so this is what try to give types of immunity. Okay, if you find the MHC1, obviously try to bind with the CD8 receptor of T cell. Okay, so likewise, okay, if you want to go for dendritic cell, the T cell activation. If you want to go for T cell activation through the dendritic cell interaction, even this kind of interaction is important. That is, the CD80 is the maturation marker of dendritic cell. Okay, if a dendritic cell is matured, okay, obviously the CD80 if it interacts with the CD28 receptor of T cell, or the T cell gets activated. So if you want to combat the cancer, if you want to kill the cancer cells, so this is the most important interaction. This is a common interaction. MHC1 interacting with CD8, MHC2 interacting with CD4 is a common interaction. Of course, these interactions classify the types of cells. It is the cytotoxic or the helper T cell. So of course, this is also an important, but the second most important, whether the T cell is getting activated or it's getting suppressed. If it is getting activated, so either you can get stimulation or suppression. Okay, there are actually two types of T cell activation. One is T cell stimulation. The other is T cell suppression. Okay, if CD8 is interacting with the CD28, you observe the stimulation, T cell stimulated. But instead of CD28, okay, it try to interact with CTLA4. Okay, cytotoxic lymphocyte antigen 4. Instead of CD28, okay, if CTLA4 receptor is there in T cell and if try to interact with CD80 or CD86 or CD83, okay, there are three maturation markers in dendritic cell. If it interacts with these markers, obviously what is going to happen? The T cell is of course getting activated, but it's going to go for immunosuppression. So if it interacts with CTLA4, you can use this T cell for treating autoimmune diseases like asthma, arthritis. So it plays a different role. You can't use it for combating cancer. Okay. So even the interaction part, whether you want a stimulation part or a suppression part, is most important. Okay, in terms of activation. So of course, if you try to find the T cell epitope, one has to find okay whether activation is happening on or suppression is happening on. So I also mentioned something about the pathway, MHC1 pathway on yesterday. Just I'm recapping you. Like if any cancer cell is coming, is getting inside the cell. Okay, what cell is this? This is a antigen presenting cell. Okay, they are mentioned like APC. APC, I already told you. The examples are dendritic cell, macrophage, okay, or B cell. So if a cell is, if a cancer cell is coming inside, of course, it's going to like generally the cell is not going to come inside, but if a cell okay is degraded, okay, or is just already killed by a cell, you can find some cancer antigens. What are cancer antigens? Of course, these are considered as foreign molecules. These foreign molecules can also enter into the antigen presenting cells. Okay, so from the cancer cells, if any cancer antigen getting inside the antigen presenting cell, example dendritic cell, it first enters into the proteasome. The proteasome majorly takes care of degradation of protein into peptides. It is just transported through the transported associated protein. Whatever peptides it generate, okay, it will be transported further in the endoplasmic reticulum. Okay, so it just try to project it on a kind of a molecule that we call it as MHC1 or MHC2. Here, if you have MHC1, okay, so generally these MHC1 molecules are just transported through the Golgi complex from endoplasmic reticulum, and then it transports to the secretory vesicle, where from the secretory vesicle it transported to the surface of the APC. So whatever MSC molecule you have, it's going to already have the epitope. The epitope is derived from the peptide of cancer. source. Okay, so whatever epitopic cancer peptide is there on the surface, on the MSC1 surface, try to interact with the T cell receptor. I told you, V alpha, V beta, two regions are there. If you try to bind, obviously you can able to find, and not only just the MSC molecule and the MSC molecule and T cell receptor, even you should find another important interaction is either CD4 or CD8. Other than CD4 or CD8, even CD80 molecule, CD80, 83 or 86 molecule of APC has to interact with the CD28 of T cell. So these three interactions are most important to get cell activated, T cell activated. If any of the interaction is not happening, like out of three, only two interactions are happened, then you can't expect the T cell gets activated. Okay. So of course, this is an important interaction, but even the other two interactions has to be done. Then only you can find the ultimately the T cell activation. Then only you can get the. So why the T cell has to get activated? If one T cell gets activated, it try to proliferate into two T cells. Okay. So during that proliferate during the stimulation, you can find one T cell developed into two T cells, and these T cells have the memory of the cancer antigen. So whenever it finds a foreign particles, okay, in the tissues or in the blood, obviously what it does, these T cells try to combat and try to engulf and try to kill even the cancer cells. These T cells directly go and kill the cancer cells and produces more proteins like this. So this is a kind of a cycle process. Okay, obviously the person will be slowly relieving from the abnormal cell proliferation. Got it? So this is the main purpose, and of course this is the life cycle of MSC, how it's happening now. And if you see the structure of MSC molecule, so the major structure looks like this. The class 1 MSC, you have here you have alpha 1 and beta 1, isn't it? But here in the class 1, you just have alpha 1 and alpha 2. Okay, so this is actually in the form of a single domain. If you see here in MSC1, alpha 1 and alpha 2 and alpha 3 are a single domain, but it is just merged with beta 2 microglobulin. Only one domain is there. But here, if you see here in the class 2, you have two domains, beta 1 and beta 2. Alpha 1 and beta 1 is majorly on the surface. Okay, so if you see here, okay, MSC1 is going to take a just small peptide because of a single domain. Okay, like alpha 1, alpha 2 is from a single domain. But here, if you see here, alpha 1 and alpha 2 and beta, alpha 1 and beta 1 are on the surface. So you should have a kind of lengthy peptide. Okay, you should have a lengthy peptide to cover from here to here. Okay, and if you see the MSC genes and function, you have we have actually two types of major genes, class 1 and class 2 MHC genes. Okay, in class 1 MHC genes, we have that is generally in proteins, we call this as major histocompatibility complex proteins. But if you call it as a genes, okay, we call the genes as HLA. The class 1 MHC genes are generally, we call that as HLA, that is human leukocyte antigen. So if you see the human leukocyte antigen genes of class 1 MHC, major genes you can find is A, B, and C. Okay. If you see in the genome, generally it lies this MSC molecules looks like this. Okay, so after B, C, and E you can find. And if you see the class 2 MSC genes, okay, the major genes you can find even from the databases is DP, DQ, DR. Okay, DP, DQ, and DR. But we also have other genes, but these are the major genes. Okay. Of course, we also have another kind of a MLC, that is class C MLC. So this is actually used for complement activation. Even myself, I'm working on heat shock proteins. Okay, during the cancer, okay, even these proteins express just because of shock. Like if a person is suddenly excited because of some shock, just like that. Okay, even these proteins can give much expression in the cancer when compared to the normal healthy patients. And if you see the class 1 MHC, more about the class 1 MHC and class 2 MHC. Class 1 MHC, of course, uh, uh, genes are present in dendritic, the APCs, which try to interact with that cytotoxic T cells. But class 2 is for helper T cells. This alone you have to remember. And difference between MNCs, you have plenty of futures. Okay, so not just the CD8 and CD4 plus T cells. If you find CD8 T cells, we call this as a CD8 plus, and here we call this as a CD4 plus. Okay, then in this case, MLC class 1, you can tell CD4 minus, CD8 plus and CD4 minus. Here CD4 plus and CD8 minus. Okay, so and if you see this co-receptors, okay, we have the different information, so which you can study even from the immunology textbooks. Okay, so the main thing which is important for computation, that alone I am teaching now. Of course, whatever I just explained you, transport associated protein, endoplasmic reticulum, okay, chaperones, all these things we can also see in the formulas. Okay, so not only we just predict the epitope based on the scores, we also go for antigenicity, toxicity, etc. For this, even we should know about the entire cycle process in class 1 MHC as well as class 2 MHC. Like, however, I mentioned you the pathway in MHC 1, even we have another pathway, a slight differentiated pathway in MHC 2. So one should understand the difference between the MHC class 1 pathway and MHC class 2 pathway to know about how these tools are predicting with the score. Okay. And this the second slide shows a few more differences of the MSc class 1 and class 2. So generally you can go for immunology by Hubei or immunology by Reut. Okay, so these are the preferred textbooks we can use to get more information on that. So if you see even the size of peptides, the textbook they have mentioned like 8 to 10 amino acid for MSc1. But for MSc2, you go for 14 to 18. Okay, so in textbooks they have mentioned like that. Okay, but uh, just because John Hopkins is proficient in this, okay, just I'm telling you, even you can go with the 13. If you go with the 14, of course, um, it also works well. And now we can go to the HLA molecules. I told you the HLA, human leukocyte antigens, are just like fingerprints. Okay, like how even the siblings can be distinguished through fingerprints. So even each and every person, the even including siblings, excuse me, can be distinguished from HLA alleles. If you see this MSc class 1, not only majorly you find ABC, okay, in the minor MSc class 1, you can also find E, F, G. And similarly, the major MSc class 2, you find DP, DQ, DR, and minor class DM and DO. We majorly focus concern on the major MSc class 1 and major MSc class 2. So about the locus information, the genes information in the human chromosome 6 is shown here. Okay, so in the chart, if chart off, okay, you can find the HLA, and you can find C and B in the long arm, as well as DPDQ and D are also present in the Q arm, long arm. So now we can see the significance of the HLA molecules. Actually, HLA allele, you can find you have numerous alleles. Okay, HLA B27 is used for to know about ankylosing spondylitis, reactive arthritis. And similarly, if you see HLA B47 is meant for this kind of diseases, and HLA DR2 systemic lupus erythematosus. So likewise, okay, even the popular diseases like rheumatoid arthritis, diabetes, we majorly concern on HLA-DR4. So if one is working on not only the cancer, and if you are focusing on autoimmune diseases, okay, you can also check this information. Okay, even for the cancers, for specific organ cancers, even some HLA alleles. Okay, A, B, C, I told you comes under MHC1. The DR, whatever DQ, this comes under MHC2. Okay, so you have to focus either MSC1 or MSC2 for these diseases. I told you IADB is, of course, the major database. And of course, you have tools developed inside to predict even the epitopes from the database. T cell epitope prediction methods. How the T cell epitopes are predicted? Actually, these are predicted based on two important methods. One is direct method and the other is the indirect method. So direct prediction procedures are based on the T cell epitope sequence and structural analysis. Okay. So not only you focus on the sequence, but you're also focusing on the structure, that is epitope structure. The 3D structure of epitope sometimes can also yield the secondary structure, which is not desired. Okay, so while predicting, we predict appropriate we use appropriate design to use these peptides to talk with the proteins. Now we can see this actually uses features like amphipathicity, motif, which is the important functional part of a protein. Okay, so the generally, if you see the functional part of a protein, we go for motif, and if you want to check the kind of lipophilic, hydrophilic. Actually, if you see the amino acids, you can classify amino acids as polar, non-polar, hydrophilic, hydrophobic. There are also subclasses of amino acids, isn't it? So if you focus on distinguishing these two types, okay, so you can go for the amphipathicity, okay. And if you see the indirect methods on the other hand predict the binding of MSC peptides by using smart techniques like ANN, artificial neural network, or support vector machines. Okay, so generally the indirect methods uses the machine learning techniques, okay, to predict the epitopes. But here it uses the basic amino acid features of a protein. And if you see the next point, the prediction of cleavage and transporter associated with antigen processing, that is TAG, are also crucial elements for the T cell epitope prediction. We generate numerous scores. Of course, we also concern on this TAP score to finalize which is the best epitope. If you see the T cell, one should also go for predicting the immunogenicity, not just predicting the T cell epitope is sufficient. We have to find the best epitope. If you see the T cell epitope, you may generate some hundred to thousands of epitopes. If you generate hundred to thousands of epitopes, the researcher, the scientist has to concern on getting the best epitope. Only one epitope he has to screen out of some hundreds or thousands of epitopes for a specific protein. Got it? So if you want to screen for the best one, okay, of course, he can also use a kind of a screening parameter. This is a kind of a virtual screening parameter, T cell immunogenicity. Okay, if you see whether the epitope is having immunogenicity or not, so that is based on the features like antigen processing, whether the antigen is processing or not, okay, by the APC, and whether this peptide binding to MSC molecules, really this epitopic peptide is binding to the MSC or not, and whether this peptide bound with the MSC is also interacting with the T cell receptor or not. Okay, all these three parameters they have used. Okay, this is a correlation of the three parameters. So whatever immunogenicity score you get from this URL is accounting for all these three features. And conservancy analysis, whether these epitopes, okay, or derived from the same source or not. Actually, it's a fraction of the protein section that the epitope contains, and identity is a correspondence. If you are taking a Nipah virus protein, for example, structural protein like W, and whatever epitopes you have designed, okay, of course, is a part of the protein, isn't it? So if it is a part of a protein, even the 9 amino acid peptide for MHC should show 100% conservancy. Okay, so whether you're getting 100% conservancy or not, this you can check from that. And also there are two types of calculation. And also one thing you should remember, even few viruses have mutations, multiple mutations. For Nipah virus, you don't have mutated viruses. Okay, you have only one strain. Okay, in 2018, whatever uh, spread in Kerala is just a single strain. But the for SARS-CoV, it is not like that. We have hundreds of strains. Okay, from 2018. So one has to focus even on the conservancy for these kind of viruses. For Nipah virus, there is no need. Okay, so this is something about the conservancy. And if you see the calculations, you have linear analysis and non-continuous sequence analysis. So linear amino acids, you just focus on the sequence. but non-linear focuses on the structure. The structure of epitope is important to determine. Okay, and not just the immunogenicity, even toxicity of a peptide is most important. If you want to give a peptide in the form of a vaccine, one has to concern on the toxicity. This toxicity is predicted by the server through these properties of ligand. Okay, the properties of the peptide. That is number one is hydrophobicity, hydropathicness. That is we call it as a hydrophilicity. Not just the account for the hydrophobicity, even the hydrophilicity is also important. We have to find the value. Molecular weight of the peptide, net hydrogens, side bulk, and steric hindrance. Okay, so considering these properties, the toxicity is determined. There is a correlation with these properties, what I mentioned. Actually, to determine the toxicity, you can use this URL, crdd dot, so what I mentioned, developed by Dr. Raghava. The tool name is Toxin Pred. And this is one such publication in the year 2020. Okay, that is a T cell epitope based vaccine for the Nipah virus. Also published one more book chapter on the human viral diseases. Now let me go to the specific article, that is T cell epitope based vaccine for Nipah virus. If you see the information of the Nipah virus, okay, it's actually a single stranded RNA virus. And if you see the size is just 18.2 kb, a very small virus, okay, but it has structural proteins like nucleocapsid, phosphoprotein, matrix protein fusion, glyco, large protein like DNA polymerase, and non-structural proteins you have CV and W. So actually, whatever I've taken is the non-structural protein W. Okay, and if you see here, how a structure of Nipah virus looks like. So you have G, N, L, P, viral. So many parts are indicated here, and even you can see how it is sequenced in the genome. Aim and objectives. Actually, the first objective is to predict the T cell epitope, and if for that you go for initially designing the T cell epitope based vaccine using immunoinformatic tool. First, you have to predict the T cell epitope. Then, whatever T cell epitope you have predicted from the ADB database, you try to perform docking. So, for docking, I used autodock. So, whatever finalized peptide I got, okay, screen peptide I got, I try to talk with. Uh, I try to talk with the HLA molecules, that is MHC molecules. Okay. So in case of T cell, I just talk with the MHC molecules. For example, whatever you got MLC1, you try MLC1 epitope, you try to interact with the MLC. Okay, likewise. To check the immunogenicity, conservation, and toxicity, we have plenty of tools in the ADB itself. Even you can use the ADB to determine all these parameters. So we also use this, of course. Of course, this should be the second objective. So, okay, so for screening this, I am also using these parameters. And then only after screening the best peptides, like top three peptides I can use for molecular docking. And finally, the best docked molecule alone is taken for AMD simulation. Okay, so this is the workflow. So that is screening the antigenic peptides, okay, through the antigen score, immunogenicity score, the other scores, whatever shown. Then whatever epitopes you got, okay, the top three peptides you try to dock, and after that only one based peptide alone you can take for MD simulation. So these are the materials and methods, IADB tools we have used, or net MSC pan, net CTL pan for MSC class 1 prediction. And for MSC class 2 server is used for class 2 prediction. So we also used immunogenicity server, MSC1 immunogenicity server for immunogenicity analysis, conservancy server for conservancy analysis. So other than this, also I told you, I have worked on B cell epitope, but for the paper sake, producing a minimum content, I just published the T cell alone. Okay, and this information, whatever shown here, baby bread and other methods or B cell epitope prediction. Okay, and toxin bread, uniprot, KB, ProPred, Autodoc. ProPred is of course a unique database. It's a separate database. It's not coming under IEDB. There are also so many individual databases, okay, indigenous databases, which shows about the MSC class 1 prediction, MSC class 2 prediction. So after talking, okay, if you want to interpret the results, infer the results, you can use Discovery Studio. And if you want to go for simulation, you can work on Chromax. This is how we have. And if you see this sequence of W protein looks like this non-structural protein. So I not just predicted the epitopes based on the net CH, net MHC and net CTL band, which is available in IEDB. Okay, also there is one kind of database called IEDB. Also, I used that. Other than these three databases, I also used an exclusive tool called ProBread. Some researchers also uses, I just use four tools, isn't it? Some researchers can also use nearly 7 to 8 tools and they screen based on that. So based on the tools comparison, may can also get a more precise peptide for targeting the Nipah virus. Okay. So if you see this predicted T cell epitopes, around 500 epitopes are generated. But I want to predict which is the best one. So for predicting the best one, I see that whatever epitopes I received, okay, is matching with this all these tools or not. For example, if you see the net MSC pan score, that should be maximum. And even net CTLA pan, if you have a maximum value, that is the best. If you see here, out of all these values, of course, 1.24 is the best one, which gives for this peptide. This peptide is best for a net MHC. But if you see here, okay, this is not good actually. This shows just zero point 0.8, okay. And I also compared few more servers also. If I compared few more servers, okay, I can able to find this peptide alone is found in most of the tools. But this peptide is present only in two tools. It's not present in other tools. This also made me to screen which is the best one for MHC1. Similarly, for the MHC2, I just screened based on the scores. Okay, so I just explain you. If you see here, you have 3.8 and here you have 1.59 for the MHC2. This 2 is for the MHC1. This 2 is for the MHC2. Okay, here you got 1.59 and 3.8. So also if you try to work with the 3 to 4 more tools, you can get an idea, okay, which is the best peptide out of 500 peptides generated. And if you see this peptide, actually, the peptide for the MSC2, I have to try with a 13 amino acid. But okay, I just tried with the 9 amino acid. But I suggest you, if you are going for a research for MSC1, you go for 9 amino acid peptide. And for MSC2, better you can go for 13 amino acid. Okay, it should not be less 9 amino acid or less than that. If you see the Nipah virus W protein, I told you, no, I not just I can't able to screen actually. If you see here, SPVI and AQPP, I can tell this is the best. But I didn't limit up to that. I also tried with immunogenicity, conservancy, and toxicity. When I tried that, okay, so of course, both are non-toxic, AQPP, and it is also non-toxic, SPVA. But if you see the immunogenicity, you have more immunogenicity. Okay, this peptide is more immunogenic. So, of course, just because of in the third server, we got this kind of a result, and even the conservancy is more. Okay, from that, we just finalize this is the best peptide. Actually, even for this, okay, using 4 to 5 parameters and filtering the best one, you can also use the machine learning algorithm. If you know the Python programming, you can use the Python programming and try to give a kind of values, okay, weightage and values, and you can also filter the base peptide from the weightage, whatever you have described. Okay, similarly, even for the MSC2, we also check for the and we finalize the which is the best one. And whatever peptides I have screened, okay, so for MHC1, this is the best peptide, SPVIAEHYY. So I just tried to talk, I talked with the Autotalk and just shown this, the peptide is talking with this protein, okay. So actually, I have chosen this kind of an allele, so which is majorly for from the Indian population. I just targeted the Tamil Nadu population. For this, I have taken specifically not just the HLA-B, but asterisk 35:03. Okay, just model this protein receptor based on the sequence what I got from the gen bank, and whatever modeled, I just try to dock with the peptide design. Already explained you in the second or third class about how to design the peptide. So for designing this kind of a peptide, one should know this sequence alone. Okay, you have to go for pepfold 3. That is the server name. In Google, type pepfold 3, and then inside that, you have to just paste this sequence. And if you click run that, obviously you get a PDB structure from the server. So you can use that for opening the 3D structure in Visualizer. But the docking, of course, we do in the Autodock, and finally, the dock complex alone is visualized in Discovery Studio Visualizer. Similarly, even for MSC2 molecule, okay, just focused on a specific population, and whatever peptide I've got screened from the immunogenicity, antigenicity, and the scores developed from the different tools. Okay, so that alone I just used for docking with the specific Tamil Nadu population. And if you see the molecular simulation, okay, just very simple protocol, okay, try it with the root mean square deviation. If you see in the root mean square deviation, okay, just take a pen. If you see here, okay, so it is almost stable up to here. Okay, so that is it's stable from nearly from 35 to 145, something like that. But later it is not stable. But if you see for a particular time, okay, it is stable over like this. Okay, find a transition of stability. So RMSD, I can tell, okay, it is stable for more than 100 nanoseconds. But even after a particular 145 nanosecond, you can find it's more stable. And similarly, the stable, I mean, how effectively this peptide is bound with the MHC molecule. If it is bound with the MHC molecule for a maximum time, then you get more immunogenicity, more concerns. So all this information is more appropriately. Okay, and RMSF root mean square fluctuations tells about the C alpha fluctuations. Here only one peak, a large peak you observe. So this alone is not stable. So if this is alone, the 100 amino acid to 115 amino acid is not stable. This alone you can check with principal component analysis. In principal component analysis, one can able to know how effectively this epitope is bound with the uh, MLC molecule or not, that is protein target or not. Okay. So again, I'm telling you, the epitopes, whatever we design, is from the Nipah virus W protein. Okay, only from the Nipah virus, we are designing the epitopes, and whatever we are the epitopic peptides derived from the viral source, we try to dock with the human MHC molecules. Got it? So we try to dock with the human MHC molecules, and whatever we have docked, we try to go for MD simulation. Okay, so we have got RMSD, RMSF, okay, and then area, okay. Even after a certain time, we can find a stable thing, okay. And how about the radius of variation? You find initially a kind of non-stable, but after some time, you can find stability. Okay, the stability extended up to 125 nanoseconds. Again, you can find unstable and you can find something. Okay, so how much stable also we can put the moment of inertia. Okay, is described in radius of variation. So obviously, I can tell that whatever score generated, I can tell even it's docked well and showing a kind of a good immunogenicity. Okay, even the T cell epitopes also play a significant role. But when compared to T cell epitopes, the scientist has proved even the similar way of predicting B cell epitopes is the best remedy for this virus. Okay, and if you see this historical, and if you see the Covishield and Covaxin, the T cell epitope based vaccine is the best one. Even in COVAX, if you see, most of T cell epitope based vaccines are used. So that is dependent, okay, whether you're going for RNA dependent virus or a DNA virus, okay, it varies even based on the structure of the viruses. Okay, now let me go to the historical development of vaccine adjuvants. So till now, whatever we've discussed is about how to design the peptide vaccines, especially T cell epitopic peptide vaccine. Now, if you want to include adjuvant as one of the component, you remember in COVAX vaccine, I told you we use nearly six epitopic peptides, and we link that six epitopic peptides with CpG nucleotide. Okay, we also link with other than CpG nucleotide, we also link with the adjuvant. Okay, adjuvant can be also a plant-based or any other biological molecule-based. If so, if you see this historical development, okay, in 1920s, okay, at that time you can find the aluminum salts cheapest source, okay, is that is inorganic metal salts are used as adjuvants. Okay, but just because of bioaccumulation in our body, so later they came up with so many organic kind of adjuvants. Even myself, I tried with AH03. The AH03, it uses a squalene based adjuvant. So for instead of squalene, a squalene derivative is withanoids, a plant steroid compounds from ashwagandha. I tried this kind of adjuvants, that is withanoids with the dendritic cell vaccines. And even the dendritic cell itself can act like an adjuvant, vaccine adjuvant. Not only the plant-based molecules, also the cell alone can also work as an adjuvant. And also I tried with the IL-2, IL-4, GMCSF. So these kind of researches happened from 2010 to 2018. So you see a lot of publications came up with interleukins, plant-based adjuvants, and dendritic cell from 2010 onwards. So this is a schematic representation diagram. I just tried in my PhD research. Okay, I've taken Poirevia diffusa. Okay, from Poirevia diffusa, I just taken a plant-based adjuvant called Poirevinon B. Where is my marker? Somewhere. Yeah, it's there. Okay, so here you have Poirevinon B. This is a plant-based molecule from. No. Oh yeah, this is a Poirevia diffusa. And Ashwagandha, I taken the Vithanol. So I just tried with two different adjuvants, but the vaccine antigens I tried is Bacillus calmette-urine, tuberculin, and tetanus toxoid. So what I do, I take these vaccine antigens, and while from the PBMC from monocytes, the immature dendritic cells are developed. So at that time, what I did, I also included this uh, plant-based drugs, okay, as a drug adjuvant. When I use this, these immature dendritic cells or got rapidly matured. So actually, immature dendritic cell develop in the fifth day from the monocytes, and the mature dendritic cell develop in the seventh day. Okay, so this is the general life cycle of the dendritic cell. But in my case, I found instead of 5 days, even on the fourth day itself, I found the immature dendritic cell, and even the maturation happened very rapidly. So even the plant-based adjuvants are used for maturation, and also it during the antigen uptake by immature disease. Okay, so you can able to find even the adjuvants play a major role in the activation when interacting with the naive T cell. The naive T cell can produce T helper 1 cell and T helper 2 cell, where T helper 1 cell releases cytokines like IL-2, IL-12, IFN gamma, TNF alpha. These cytokines can interact with the cytotoxic lymphocytes to generate effective effector cytotoxic lymphocytes. Whatever you generate, okay, so this will directly go and combat the cancer. Not only this go and combat the cancer, even the cytokines released TNF alpha can also directly go and combat the cancer. And even if you see the naive T cells, so which also produces helper 2 T cell, this will try to interact with the B cells. Okay, if it interacts with the B cell, it generates two types of cells, plasma cells and memory B cells. Whatever memory B cells you generate, this also try to go to combat cancer. And even the antibodies generated also have a kind of a memory which can also try to go and combat the cancer. So there are numerous cells and numerous cytokines, okay, or including antibodies, try to target cancer indigenously. if you see the adjuvants, okay, there are so many kind of adjuvants in the SARS-CoV-2 vaccines itself. If you see in mRNA spike protein, okay, Pfizer, they have used polyethylene glycol, which is 95% effective. And AstraZeneca, okay, the coronavirus vaccine A, that is AZ D122, okay, which uses the adenovirus, can give 60 to 90%. And Covishield, okay, developed by the Janssen, so CD4 T cell response, 70% effective. Likewise, Covax, okay, Sputnik. So almost all the things, okay, all the vaccines uses the adjuvants. It can be a viral vector, okay, even the viral vector can act like an adjuvant, or even the aluminum salts like aluminum sulfate or aluminum phosphate, which is the cheapest adjuvant sources. Also, they are using this matrix M adjuvant where they are used nanoparticles. In COVAX, they used 40 nanometer nanoparticles of saponins. Saponins are the plant-based compounds. Okay, so these are used there along with the cholesterol and phospholipid. Why they use cholesterol and phospholipid? This is a effective drug delivery. This is an effective kind of vaccine delivery. So like if you want to slowly release the antigens and to if you want to produce antibodies for a maximum period, then you can go with a combination of adjuvant cholesterol and phospholipids. So likewise, there are so many kind of an adjuvants used for the not only treatment of the viruses but also for the treatment of cancer. Myself, I also developed an adjuvant immunoanalytical database, okay, from the C-sharp, SQL, and PHP, okay, for the plant-based adjuvants for the treatment of autoimmune diseases as well as cancer diseases. Okay, of course, PG students helped me to develop a kind of a database. And related to the hands-on experience. So now I'll be taking you to the IEDB tool and I tell you how to predict it. Before that, one should know about the Nipah virus W protein. So first, we can take the source of Nipah virus W protein. For that, I'm getting inside and searching for Nipah virus. If you know the FASTA code, it will be easy for me to fetch. Actually, I have a code. Okay, this is the Uniprot ID. Okay, this is the Uniprot ID for the Nipah virus W protein. I've already searched, so simply I'm typing the ID, click search. So initially, the first link, whatever shown, okay, I think that directly is taken to the protein W of the Nipah virus. Okay, so you have to take the sequence alone, isn't it? So go to the sequence. If I click the sequence, okay, if you go for the Unipark, you can get the uh, format, okay, in Unipark. Unipark, show sequence. Oh, it shows only this. I can take a sequence like this. Okay, I take the sequence, or even I can download the sequence. FASTA format is there. I've downloaded the sequence. So this is my FASTA format. What I have to do, I have to go for initially MHC1 binding in T cell epitope. Okay, so here I'm going to paste it. Okay, paste the what they're telling entire protein sequences in FASTA format. Okay, if you have want to go for multiple protein sequences, use a space and give multiple sequence in FASTA format. Either you paste it or you just upload the file. Okay, the file name you have to remember that it should be file name dot FASTA in double quotes. If you save it in Notepad, remember you will be saving it in text documents file format, but file name .fasta in double quotes, or it just simply paste it. And now it's asking what exact database you want to use. Defaultly, it uses Net MSC Fan 4.1, which is recommended, but also we have other databases. These are other databases you can use, but I'm going to use this. Actually, this is a prediction method. Okay, this is the prediction method. I selected like this, and now it's asking for the alleles. So we don't know about the alleles, isn't it? If you want to select a specific population, how can we make it out? So before running this tool, I suggest you to go with population coverage. Okay, you know, where is population coverage? It is an epitope analysis tools. In the population coverage, okay, you have a select area of the population. So we have to select India, isn't it? Any other thing we have? We also have allele frequency net database. Better you can go to this. So instead of the population coverage, I'm going to the allele frequency net database. You find a hyperlink. I'm directly go because here you get just the India Asian. But I am not just want to get for the Indian population, but I want to get for a specific community of a state. So I'm taking you further inside. Okay, now I have to click in the allele frequency net database. You have to click populations. So in the populations, you have an information for specific geographical regions. You browse the populations by geographical regions. Now it is getting insight further in a specific region. Okay, so we want to focus for India, isn't it? The India lies in South Asia, I think. South Asia and what else? Southeast Asia. I can check it out. Whether we found in Southeast Asia. No, you find more China. I think we are in South Asia. So if I click South Asia, yes, you got. See, for Indian alleles, you have for India, Andhra Pradesh, Brahmin, Kolla, Sunni. So many specific community people are there. If I want to focus on the Tamil Nadu, Chennai population, Dravidian, even you can screen it. So what you can understand, if you focus on Tamil Nadu, Chennai, okay, the major allele you can find for in Chennai population is the MHC2 allele. What is DR? MHC2. If you find DP, DQ, DR, obviously comes under MHC2 allele. Here you find majorly this is the common one. But we don't find any MHC1 allele, isn't it? Even if you want to retrieve an MHC1 allele for a Tamil Nadu population, just see down. Okay. For an order, you have A, B, C. But if you go up, India, Tamil Nadu, you have A, B, C, DQB1 and DRB1. Okay. For example, if you want to focus on the Chennai, simply click Chennai. Now, inside the Chennai, okay, you get this information, DRB1 alone. But if I want to go specifically to Tamil Nadu, Chennai, you find DRB1 01, 03, 04, 07. See, nearly 15 alleles are there. 15 alleles are there, okay, in the Tamil Nadu, Chennai. So you can use this for the prediction. But now presently, I am focusing on MHC1. I am not focusing on MHC2. Okay, so better come out. You can focus where you can find the MHC1. So for this, I'm going to the NADAR community. Inside the NADAR community, allele frequencies. In the allele frequencies, you can find 33 alleles. Okay, you find HLA allele, B allele, C allele. Almost all the HLA alleles are there in the NADAR community. For example, if I want to focus on first two alleles alone, because I don't have time to take all the 33 alleles, just take 0101, 02, 01, 01. So for this, okay, 0101, and the next one what is shown is 0201, isn't it? Somewhere where I missed 0201. So I'm going to this first 0101 is selected, isn't it? The length I have to take it as 9. Okay, after filling this, we found this as this entity is saved. Now 0201. For this also allele length 9. So give me some 4 minutes. Okay, I try to complete it. And the email ID, I give some email ID, okay, to generate this course, okay, to generate the results and save a copy to my email ID. You'll be getting results within a minute. So, guys, see, immediately got the results. But whatever results you got, okay, you got plenty of results. So, if I drag from the top to bottom, see this much results. So, these results can also be downloaded from here by clicking this hyperlink. So, you will be getting an excellent data which can be opened in Excel sheet. Okay, this can be opened. But if you see which is the best peptide predicted, okay, according to this tool, the top one which shows the maximum score is the best peptide. That is, this is the best peptide for MNC1. For Nodar community, if you want to design a vaccine for the Nipah virus, especially if you want to target W protein, of course, this is the best peptide. Okay. Similarly, you can go for MLC2. For MLC2, you can go for MLC2 binding. In the MLC2 binding, again, it's asking to paste one in the FASTA format. Let us see whether I have. Yes, I have the FASTA format. I pasted, no format detected. Why so? Uh, FASTA format detected now. Okay. So try to remove the space. This gives you even a clarity whether you're working perfectly or not, or else we're not sure whether it's considering gap. Okay. However, I just completed, and then now it's asking for the null. You remember for MLC2, you have to go for 13 amino acid, isn't it? But you don't know what you have to select. For example, for the Tamil Nadu Chennai, you choose the DRV10101, then you select this 0101. And if even if you find 0102 is significant, you select this. Okay, and the size you select is the 13. Okay, so this alone you have completed. Now use your email ID for better saving your results. Okay, now even select someone alleles HRF, or you can select whatever you want. Simply click submit. So here also get a table of results. Okay, for MHC2. So for alpha T cell and for cytotoxic T cell, you have generated the best peptides. So whatever peptides are generated, you can see this is the best 13 amino acid peptide. Okay, so the top score, okay, whatever the maximum score gives the best peptide. So this is the best peptide generated for the MHC2. Got it? Even the results can be downloaded here. Okay, by clicking here, the same thing you can get. Okay, so nearly thousands of epitopes are generated. Not even 100, thousands of epitopes are generated for the W protein. So now you have learned how to go for MSC1 and MSC2. There is also tools to go for MSC1 processing. Okay, MSC1 processing. So in MSC1 processing, you have to click this. Other than processing prediction, you have neural network based prediction for proteasomal cleavage sites, net chop, T cell epitopes ka NetCTL and NetCTL pan, MHC and P, prediction of peptides natural process. So we have these tools also. If you want to predict the immunogenicity, click this. We have plenty of tools. Only thing is that you have to paste the FASTA format. Simply pasting the FASTA format is sufficient. And you can give, if any length is asked, you can provide, or it's just like that, just keep it up. Okay. But you can provide even email ID so that you can aware of what exactly sequence. Oh, entire peptide sequence. It's asking one is the peptide. So initially you have predicted peptide one, isn't it? Where is that peptide? Yeah, this is the best peptide I predicted for MLC1. Okay, this is the best one. So it's asking only to give the peptide to calculate the immunogenicity. First, I pasted the peptide one, and second peptide, I'm going to take this, the immediate peptide. So I paste some series of peptides, okay, not the sequence, just the peptide sequences you have to give as an input. Okay, they're asking use the comma separated markers. Comma, sir, okay, comma. Okay, I gave like this. Okay, click submit. The tool has been integrated, next generation tools contains an invalid character. Okay, I give this. What happened? Oh, at position 10. Okay. Yeah, we got the result now. Okay. This shows the best score 0.12082. When compared to this is the best one. QL. Where is QL? QL is shown as the second best here. But when you've gone for the immunogenicity, it shows the best one. So like this, I suggest you to go for the top 10 base peptides and check the scores, create the score based even on the immunogenicity. Like immunogenicity, you can also predict the toxicity. Okay, participants. Okay, thank you for your patience. And if you have any doubts, you can ask me. NADAR community database is taken from, yes, it's from the government, of course. It's a public domain database. Okay. It's a this is what we call this as a immunoinformatics database. Any other doubts? Okay, if no doubts, thank you for your patience and interaction.